Good morning and welcome to the class. So in our last class we started learning about heredity and uh, how Mendel worked with pea plants, how he studied about the monohybrid crosses, the dihybrid crosses, how the genetic factor actually goes from one place to the other place, from the parents to the offsprings, and how they are responsible to give that characteristic trait. We saw that some traits are dominant whereas some are recessive, right? And not only we checked about the dominant and recessive, we also saw that some of them are incompletely dominant, some of them are co-dominant, and yeah, we also saw that there is a relationship between the genes which are next to each other. We call that as linkage. Yeah, not an exact relationship, but we see that when they are very close to each other, the recombination is very low, whereas if the genes are really situated far away in the chromosomes, we see that they are the recombination rates are higher. So we have studied about all these aspects of how the traits are passed on from one generation to the other. Now, if that is what we know, the next part that we would really like to know is what exactly is this gene. We know that there is this gene, the DNA, that is present in the gametes, whether it is the egg or the sperm. Now, we would like to know how this DNA exists in each cell, why that information is stored in the form of DNA, and if there is a DNA, right, our DNA is not giving me characteristics. Obviously, say I'm talking about a red color flower, the red color comes not from the DNA but from one of the pigments, which is a protein, right? So all the traits are expressed in the form of proteins and not from the DNA. So there has to be some mechanism where the DNA kind of reads the message and reads it as a protein, right? So we are going to study about that particular aspect in this chapter, which is called as the molecular basis of inheritance, right? We are interested about what exactly is DNA, what is the nature of DNA. Of course, the structure of DNA we have already studied. We will also be studying that in chemistry part. Okay, in biomolecules we'll be studying that. Here we are going to study with respect to biology. We'll not go too much into the chemistry aspects there. We also study about how that DNA will replicate during cell division. Okay, not during cell division, it is in the S phase, how the DNA doubles. We also study about how this DNA is converted into a protein or reads it as a protein, and what controls this gene expression. After studying all this, we will talk about one very interesting part of this whole molecular biology, which is called as the Human Genome Project, where we have taken all our cells and try to sequence it, try to find out what is the sequence, what is the DNA that we have, whether the DNA codes for any protein or it is not coding for any protein, and so many other aspects. How many alleles are there for a particular protein? So all these are being studied in a massive project that started in 2000, which is called as the Human Genome Project. And after we learn about all this, we talk about one application part of learning all this DNA, which is helping us to find out, how, if I know the sequence, what are the uses of that? Okay. Anyway, I can't read what is ATGC in my own body. So what is the point of finding out the sequence? There are many uses for that, and we use one technique to do that, which is called as the DNA fingerprinting. So in this particular chapter, we are going to talk about the innermost, very beautiful, intricate aspects of how the information which Mendel studied, about the factors, works in each and every cell. Okay. Yes, here we are not concentrating only with respect to the gametes. We are not talking only with respect to the reproductive system or with respect to evolution. Here we are trying to understand how every cell behaves, how every cell interacts, and how every cell divides. Okay, so that is what we are going to learn in this beautiful chapter, which is called as the molecular basis of inheritance. Uh, so before we start off talking about the molecular basis of inheritance, I would just like to tell you that the whole science, the molecular biology, which we call, or the molecular basis for cell, all this started as science only after, you know, the Hershey-Chase experiment, which proved that DNA is the genetic material. Okay, then there were a lot of experiments in the field of biology trying to find out where this DNA is, how it behaves, and many other aspects. In fact, whatever we know till now about our DNA is, they say, in the infancy of the field of biology. We do not know much about it. We have our own theories to state, we have our own understanding of whatever knowledge we have, but we still do not know whether all that is true, because there's lots and lots of things that has to be done in this particular area. Now going further, we also see that there was a massive growth in technology. When there was a massive growth in the technology, we were able to use all that techniques and the technology to study the smallest particle. Say I wanted to isolate the viral DNA. Yes, it was possible because we had an ultra centrifuge. Then I had to see the DNA, visually find out whether it is there in my extract. I could do that because by that time I had say UV spec or something like that. Therefore, I could use a lot of gadgets to really find out these fascinating things that's present inside my cell. Right. So with these two important factors, the whole of molecular biology took a start, and right now, yes, we know much about it, but that's not enough. We have much more to study. However, we will start off learning about what is known to us, the whole of molecular biology or the molecular basis. We go to the things that we cannot see as major molecules. Say for example, I take a cell. I'm sure you have studied about the cell. You know the structure of a cell. Now when you're taking the structure of the cell, you know that there is nucleus, there is DNA, then there is mitochondria, then the chloroplast, and so on. You also see that there are so many proteins present. Now when we are talking about the molecular basis, we are not talking about the cell, but we are more interested in trying to find out the molecules that are present inside the cell. We will talk about how these molecules are synthesized. We will learn about how these molecules are getting originated and what is that controls the origin of these molecules inside the cell. So these kind of aspects is what we study in molecular basis. Now we have done enough study and we have realized that for understanding any molecular basis, the most important substance which is present inside the cell is the presence of the nucleic acids. Now when we are talking about the nucleic acids, we know very well that there are two types of nucleic acids. One which we call as the deoxyribose nucleic acids, and the second one is ribose nucleic acids. Many of them write it as ribonucleic acids. So ribose, ribo, anything would be fine. Ribose nucleic acids. Very commonly we call this as the DNA, and this is called as the RNA. RNA is DNA is the genetic material in most of the organisms. You take any bacteria and any organism other than bacteria, higher than bacteria, you see that all of them would have DNA as the basis, as the most important genetic material. RNA is also a genetic material, but yes, it is not present as genetic material in all the places, in all the organisms. RNA is present as genetic material in very few viruses. Okay, we call them as RNA viruses. I'm sure you have studied about the AIDS virus and HIV virus in your 9th and 10th standards. Yes, that is a RNA virus. Flu virus, yes, that's a RNA virus. Retroviruses, so there are very small group of viruses which have RNA as the genetic material. Many many viruses also have DNA as a genetic material, but when we talk about any other prokaryote and eukaryote, we see that DNA is the genetic material. So, do we see RNA in our cells? Yes, very much. Okay, RNA not only serves as a genetic material in few organisms, RNA also behaves as carrying some information. Okay, something like a messenger. So we have some kind of RNA which is called as a messenger RNA. Okay, very commonly we call that as the mRNA. Now this mRNA is present in our body. There is one more RNA which kind of acts like a plug. You know, it makes this mRNA which is carrying the message to bind to the ribosomes. Ribosomes we have already studied, they are the protein factories, right? So protein factories should come in contact with the messenger RNA which is carrying the message. Now these two have to be together, so there has to be some kind of plug, right, or an adapter. Now such kind of adapters are also present in our cells as the tRNA or the transfer RNA. Okay, they kind of help the amino acids, then the mRNA and the ribosomes, all of them to be together. We call that as the transfer RNA. So we will study about the importance of this tRNA and mRNA in detail later. Okay, some RNAs, you don't believe that they also behave like catalysts. Okay, we have already studied that all the enzymes, you know, are proteins. There are some catalysts which are actually RNA in nature. Okay, so we have something called as siRNAs, then we have something called as the miRNAs, and so many more, and all this is very important for our understanding. Okay, all this is very important for our understanding of how the cell really works. Okay, when I'm telling you about transcription, translation, the central dogma, how the DNA gets converted or reads it as a protein, then you see that the RNA in our cell plays a very important role. Otherwise, RNA is not a genetic material, but yes, it is very important for us to carry out various other activities. Okay, so this particular chapter we will start off learning about the most important nucleic acid which is carrying the entire genetic information with us, which is called as the DNA. Now DNA is basically deoxyribonucleic acids, right? Now DNA, what exactly is it? We know that it is a polynucleotide, right? It's a nucleotide, nucleic acids, right? Nucleic acids, so we call it as a nucleotide. Now what exactly is a nucleotide? What exactly do we mean by a nucleotide? Now whenever I am talking about a nucleotide, you should know that this particular nucleotide is a component which has three important regions. It has three important regions. The first important one is a pentose sugar. This is not for DNA, this is the common thing that I'm telling for polynucleotides. So in the case of a nucleotide, you will see one pentose sugar. Okay. Now in the case of DNA, it is deoxyribose, whereas in the case of RNA, it is the ribose sugar. So our DNA has the sugar which is deoxyribose. Our RNA has the ribose sugar. Then the second important thing that is present in a nucleotide chain, in a nucleotide, is something which is very important, which is called as a phosphate group. What is a phosphate group? Phosphate group basically is like a link. Okay, what does this do? It keeps all the sugar molecules attached to one another. Okay, so there are many sugar molecules, all of them which are attached to one another, and because there are many sugars attached together by a phosphate group, we call this as a polynucleotide. Okay, this poly comes because of the presence of the phosphate group, because this is the one which helps it to make it into a poly. Right. So polynucleotides are formed because many sugar molecules are linked to each other with the help of a phosphate group. Good. Now there is one more component that is present in a nucleotide, a very important component, that is the one which kind of gives you the characteristic of being unique. Okay, and that is something that we call as a nitrogenous base. What do we call this as? We call this as the nitrogenous base. Now when I'm talking about nitrogenous base, a nitrogenous base is basically a kind of ringed structure, right? It's basically a ringed structure. It has nitrogen present in that ring, and we have two such rings. Okay, two such bases. The first one is called as the purines, and then we have the second one which is called as the pyrimidines. So there are two rings here, there are two types of nitrogenous bases, purines and the pyrimidines. Now please remember that we have two types of purines and three types of pyrimidines. Purines, we have two of them, which are called as adenine and guanine, and then we have pyrimidines, which are cytosine. Then the second one which I would write is the thymine, and then we have a uracil. Now if our nucleotide has all these three, you see that because of the phosphate group, it has the ability to bind to many sugar molecules, and hence we have a polynucleotide chain. Okay, therefore I can call this DNA as a polymer of deoxyribonucleotides. Yes or no? So this is a polynucleotide, has a deoxyribose sugar, and it forms a polymer. So DNA is nothing but the polymer of deoxyribose nucleotides. Now let me tell you here, how long is my DNA? I have to measure it, right? Now obviously I can't measure it with so many millimeters or centimeters, that is not possible. So we measure it in the number of bases they have. Okay, how many bases they would have? So we know what is a base now, we know that it has a nitrogenous base. So how many bases are present in a nucleotide chain? Okay, that would be the length of the polynucleotide chain. Say for example, just to give you an example here, there is one small virus which eats a bacteria. We call that as bacteriophage. Okay, bacteriophage lambda, then we have bacteriophage 5 and many others. Okay, if I'm talking about bacteriophage lambda, you know how big a bacteriophage would be. It's a small virus. Now that small virus bacteriophage lambda has a DNA which has 48502 bases present. Okay, actually basis is not the right term, but I will tell you in some time about one more thing there. Okay, so we have 48502 bases present in that polynucleotide chain of bacteriophage lambda. Then we have E. coli. Now in the case of E. coli, it has 4.6 into 10 to the power of 6 base pairs. Okay, it's called as base pair. I'll tell you what a base pair is later. Okay, if that is the content there. Now in the case of humans, okay, the haploid content. So in the sperms and the eggs, the DNA that we have is somewhere around 3.3 into 10 to the power of 9 bases. So you can imagine how long our DNA could be, right? So in the case of E. coli, it's a circular DNA. In the case of lambda phage, again it would be a kind of circular. Whereas in the case of humans, you see that they are all divided into chromosomes. So the whole content of DNA would be somewhere around 3.3 into 10 to the power of 9 base pairs. So in a diploid condition, you can do the calculations there, right? So this is how our DNA is. Okay, but we should not stop at this, right? We should study more about our DNA. So what I will do is I show you this particular structure. I hope you can see this. Yeah, looks pretty clear. So how exactly all these are joining? You see, this is basically a pentose sugar. Okay, you can see that there is 1, 2, 3, 4 and 5 sides. So there are 5 carbon atoms here. So you call this as a pentose sugar. So this is a deoxyribose sugar. Now you might be wondering what exactly is there here, how many carbons, how many hydrogens and how they are linked to each other. Yes, that part I will show you in the next slide. But you see that there is a sugar molecule, there is one more sugar molecule. These two are linked by the phosphate group. This sugar molecule, there is phosphate group. So this keeps on moving. Okay, this keeps on not moving, this keeps on growing like this. And at this position, which is the C5, okay, at the C5 position, there is a phosphate group. At this C5, you have a phosphate group. At this here, you have a phosphate group. Please do remember that this 5' phosphate, or this at this particular position, there is no attachment of one more sugar, right? So at this 5th carbon, you see that it is ending here. The DNA polynucleotide chain ends here, and this position where you have only free 5' free 5th carbon atom free, right? You call that as 5' phosphate. What do we call that as? We call that as the 5' phosphate group. Okay, please let us see here what is happening. Now in this position, you see that there is the third carbon atom is attached to this, right? So you see that there is the third carbon that is free here, and always here there will be one OH group. Okay, there is one OH group, and that we write it as the 3' OH. So basically, if I take any DNA, there is one lower part and one upper part. So one of them will have a OH group and one of them will have a phosphate group. So we call this as the 3' hydroxyl group and the 3' phosphate group. Now just look at this structure here. Now this is the structure of the ribose sugar and the deoxyribose sugar. Now in the case of the ribose sugar, we have oxygen and we have carbon 1, 2, 3, 4 and 5. Now carbon 1 has OH and H very much similar to what you have learnt last year. We have learnt about all this. So then you have carbon attached to hydrogen and OH, whereas in the case of DNA, you have the deoxyribose sugar which lacks this oxygen. Hence the name deoxyribose. So in the second carbon, there is no oxygen. Yes, so hence the name becomes deoxyribose. Okay, so you see that there is no oxygen here. So you have carbon attached to hydrogen and hydrogen. So this is the sugar that we have in our DNA, right? And then we have the third prime. Now this third prime oxygen, sorry, this third prime carbon has OH and H. Now what we see is when there is a phosphate group that is attaching, this H comes out and that phosphorus group gets attached here. Okay, so we have oxygen comes out, so you have C, H and H, and then the phosphate group attaches here, and then you see that it keeps on growing, right? That is the reason why if there is one end with free phosphate, the other end you will have the free OH, because this OH is not participating in forming the nucleotide, right? Okay, so then we have the fourth carbon atom, which is attached to hydrogen on one side, and then here you have the CH2OH. Now here again you see that there can be a phosphate group that is present. So this way we have the sugar structure, the ribose sugar structures, which are very, very important for that. Okay, many times even before the addition of nitrogenous base, you see that there is addition of before the addition of the phosphate group, you see that there is addition of pentose sugar with the nitrogenous base. Okay, so what would happen there? So there you see that there is here in this region there is attachment of a nitrogenous base. Right? Now such compounds where there is a ribose sugar or the deoxyribose sugar attached to a nitrogenous base, we call that as a nucleoside. Okay, what do we call that as? We call that as a nucleoside. So basically, first thing that you see in our cells is that we have the sugars which are attached to the nitrogenous base and they form the nucleosides. So these nucleosides have to be given a name, right? So suppose there is cytosine attached to say deoxyribose sugar, we call it as deoxycytidine. If it is attached to guanine, we call it as deoxyguanosine. So depending on where it is, how it is attached, we call it as a different name altogether. Yes, so I hope you are understanding what I was telling you here. So there are two types of sugars, the ribose sugar and the deoxyribose sugar. Both of them very important to carry out various functions, because the sugar is such that it can bind to the nitrogenous base, it can bind to the phosphate group in these two positions, right? Thus you see that there is the structure of the ribose or the deoxyribose sugar is just apt, right? Okay, now comes the next part of this. This whole chain gives rise, okay, let me just tell you about this and then I'll tell you about that. So I told you about the purines and the pyrimidines, right? So purines are adenine and guanine. You can see that there are two ringed structures basically with nitrogen present in them, so we call them as the nitrogenous bases. And then we have the pyrimidines again which have nitrogen present in them, but you see they are single ringed structures, so I call them as the pyrimidines. Now just to tell you one interesting fact here, though we have purines and pyrimidines, both of them in our polynucleotides here, you see that uracil is present only in RNA. Okay, you cannot see uracil in DNA, and at the same time thymine is present in DNA only and not in RNA. So this is something that we see in our nucleotides. Okay, so how, if I give you a polynucleotide and I ask you to find out if it is a DNA or RNA, you should just look if it has uracil or thymine. Okay, if there is uracil, it is a RNA strand. If it is thymine, it is a DNA strand. Okay, in fact, just to tell you about evolution here, we earlier had questions saying that why can't RNA be our genetic substance, because you see the structures, both are polynucleotides, right, and almost the same components other than thymine and uracil, that everything is the same. But if you just look deeply, compared to thymine, uracil is unstable. Okay, this presence of CH3 group gives lot of stability to thymine compared to uracil. Otherwise the structure remains the same. You see that all of they have the same structure. This presence of CH3 makes all the difference. The CH3 gives the whole of thymine lot of stability. And since our genetic material has to be something which does not change very often, right, I think nature chose DNA, the one which has thymine, the more stabler nucleotide, nucleonitrogenous base, to be a part of our genetic material and not RNA, right. So this is about our structure of purines and pyrimidines. Now just like as I explained to you how they would form a bond, I'll just explain to you using this particular diagram. So we have, let us just start off from here. So we have the first sugar here, deoxyribose sugar. See that oxygen is missing here, so no oxygen, so this is a deoxyribose. Then you see to the carbon 1, we see that there is attachment of a nitrogenous base. Okay, that is a nitrogenous base, it's a single ring, so it is a pyrimidine. Then you have the fifth carbon atom, which has CH2OH, right. Now this CH2OH is linked, that OH is replaced by a phosphate group. Phosphorus attached to 4 oxygen atoms here, so you have this CH phosphate group, which is called as it forms a phosphodiester bond. Okay, so this bond is formed, and it is formed when there is one hydrogen loss from this second sugar atom here. So one hydrogen from here and one hydrogen from here is lost, and we get a bond here. So C5 of one sugar and C3 of one sugar, the other sugar, will participate in the formation of a phosphodiester bond, right. So this is how it forms. So this so then we have another CH2 here, O here, and the one more phosphorus group which attaches to the C3 of another sugar, and the chain keeps on growing that way, right. So this is how we see the formation of a polynucleotide chain with the help of the phosphate group, right. And then we also have some nitrogenous bases here. So you have a pyrimidine, purine, and purine, right. So we have the pyrimidine bases and the purine bases, not that it's 2 after 2 and all that. It can be in any combination, which I'll tell you in some time. So this is how our DNA is polynucleotide chain. But now comes one very interesting and important part of it, and that part is something where you see that our DNA is not a single strand. It's not that you have one polynucleotide chain here and yes, this is DNA. No, not at all. Okay, it is not at all going like this. You see that there are 2 nucleotide chains. Okay, so there are 2 nucleotide chains, and those 2 nucleotide chains come and form a bond. Okay, so there is one nucleotide chain here, there should be one more nucleotide chain in this here, and yes, when I'm talking about two nucleotide chains, there has to be something which holds them together, right? And that holding of two strands of the polynucleotide chains, okay, two strands of the polynucleotide chains, they are held together by the interaction of the nitrogenous bases. So we have one purine here, purines here, pyrimidines here, and so on. Now these will interact with the purines and pyrimidines of the other chain that would be present. Now when I say interact, it is not talking, it is all by the formation of the bonds. And when we are talking about the bonds, please do remember, we are talking about a genetic material which has to be stable, which is able to carry the information, and at the same time, when the cell really wants, it should be able to open up, read the code, and read it in the form of a protein. Yes, so I want it to be stable, at the same time I want it to be flexible. Therefore, the bonds that are formed between these nitrogenous bases is so beautiful that you see that they exist in the form of hydrogen bonds. Okay, they always exist in the form of hydrogen bonds. So suppose I have thymine on one side. This thymine is superb molecule, has this superb memory. Its structure also helps to do that, and you see that it can bind only with adenine. When there is thymine on this one strand, there has to be adenine on the other strand, and that is how it is made. Yes, that is how it is made, and therefore you see that whenever there is thymine on one one polynucleotide chain, and adenine on the other polynucleotide chain, there is some kind of affinity between them. They come together, and when they form a bond, you see that there are two hydrogen bonds that are formed between them. So A pairs with T. Beautiful, right? Similarly, we have cytosine always pairing with guanine. Guanine is again a pyrimidine having two benzene ring structure, and cytosine having one of them. So you have G pairing with C. You see that there are three hydrogen bonds. So in a polynucleotide chain, and when we are talking about the DNA, we have two polynucleotide chains, and these two polynucleotide chains are held together by the hydrogen bonds formed between the nitrogenous bases. And these nitrogenous bases are so specific that always A pairs with T and G pairs with C. Right? So this is the one, and then on the backbone we have the sugar phosphate backbone. Okay, so the sugar moieties, the phosphate moieties, they all are the backbones, whereas the nitrogenous bases and the hydrogen bonds that are formed between them is the one which keeps these two polynucleotide chains together. I hope you got what I said. Okay, now talking about RNA, now RNA is slightly different. Because of the presence of the OH group, you see that RNA is slightly having only a single polynucleotide chain, and it does not form a two polynucleotide chain. That double helix is not formed. However, you see that many times the RNA can form a loop on itself and give rise to a hairpin structure and all that. But whatever said and done, DNA is not double stranded. DNA is single stranded. RNA is single stranded. DNA is always double stranded. And yeah, at some points you see a double stranded RNA, but we do see that in eukaryotic cells, double stranded RNA is quite unstable, and yes, it cannot be on its own for a long time. Okay. Now, basically, this whole of DNA, is basically acidic substance. Okay, and this was first identified as an acidic substance present in nucleus by a scientist named as Frederick Mischer sometime in the year 1869. Okay, now because this particular acidic molecule was present inside the nucleus, he called this as nuclein. Okay, nuclein. But the problem was, I was telling you that the technology was not that great. Okay, now isolating this particular DNA was not at all easy. Okay, it took a very long time. I guess we will study this later in detail in biotechnology chapter, where we get to know that though this was identified in 1869, the whole extraction and study of its structure, the whole of crystallization of this particular molecule and studying its structure was done in the year 1953. Okay, so there were many scientists like Wilkins, and then there was some lady called as the Rosalind Franklin, Watson and Crick. They kind of contributed a lot to understand the structure of DNA. Right now, once they studied that structure in the year 1953, they kind of studied about the X-ray diffraction studies, crystallization studies and many others, and they found out that DNA is not a single polynucleotide chain, but it has two polynucleotide chains, and these two polynucleotide chains are kind of spiraling. Okay, and this spiraling DNA molecule was called as the DNA double helix. It was called as the DNA double helix. I'm sure you have seen that spiral ladder. Yeah, this looks very much like the spiral ladder. Okay, so one going next to the other, it's kind of spiraling. Okay, then 1953 they proposed this, and then there was one more scientist. Again, it would be wrong if we do not get to know his name, who is called as Erwin Chargaff. He was the first person to find out that adenine and thymine get paired, guanine and cytosine gets paired, and always, that is 3 bonds in GC and 2 bonds between A and T. And he also said that, the number of adenines will be equal to number of thymine, number of guanine is equal to number of cytosines. Okay, that so the ratios of A's and T's, G's and C's are always constant, and it has to be equal to 1. Now this was proposed by Chargaff, and in fact it's called as the Chargaff's rule. Okay, so this binding of adenine to thymine and guanine to cytosine, they are very specific, right? Now we call that as the DNA complementarity. Okay, we call that as the complementary nature. DNA complementary nature, and again very important, because if I know sequence in one strand, I will get to know the sequence in the other strand. Yes, in fact this is the basis for how the DNA replicates and transcribes and so on. So most of the functions of the DNA as a genetic material is attributed for its complementary nature. Okay, so this is about the DNA. Now let us just study about the structure of the DNA. I'm sure you are able to see the structure here. Talk about the DNA structure. Now this basically is a helical structure. Okay, DNA is basically a helical structure. So you see that it is helix. It has two polynucleotide chains, which we have already seen that, right? So there is sugar and phosphate backbone. The one which you see in the light blues and the dark blues is the sugar phosphate backbone. And what is inside this spiral structure is our nitrogenous bases, right? So basically it would be something like, you have, if you can imagine yourself with a staircase, okay, a ladder. So you see that this would be sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. So it would be something like this. So this would be the you know strands there, and then the nitrogenous bases would be the stairs of the ladder. Do remember that there are some hydrogen bonds here, 2 or 3, depends on whether we have AT or the GC. Okay. Now the two bases in the strands are always paired by the hydrogen bonds which are present. Adenine with thymine, 2 bonds, guanine with cytosine, 3 bonds. Okay, so that adenine can be in this strand or this strand. If here is adenine, here would be thymine. If there is thymine here, then there would be adenine there. So it's like, complementary nature. So you see that adenine and thymine will always bond with each other. Now comes one more interesting things. Have you noticed in this particular diagram that if one is a purine, the other one is a pyrimidine, right? One is purine, the other one is pyrimidine. And if you just go back, yeah, you see that both the pyrimidines have 2 rings and all the purines have 1 ring. Now if there is a bond between a purine and a pyrimidine, you see that the distance between the 2 strands always remains the same. It is like this only. It is not that my ladder looks something like this. My ladder does not look something like this. It doesn't look like this, because the nitrogenous bases occupy the center position such that there is nitrogenous base, hydrogen bonds, another nitrogenous base perfectly, okay, and that diameter is 20 nanometers. You take bacterial DNA, you take human DNA, you take tiger's DNA, all of them, they have 20 nanometers. And you know the best part of this? The best part is this part of the DNA, we call it as the bDNA. bDNA, it should have the right amount of water present in the cells. Okay, then we call that as the bDNA. There are many other forms which we will learn in biochemistry. Anyway, talking about this, we have the bDNA, and you see that the chains are coiled in a right-handed way. Okay, you see that they are coiled in the right-handed fashion, and this distance is 34 nanometers. Okay, and this one would be 3.4 nanometers. Okay, one of the you see that the pitch of the helix is 3.4 nanometers, and in one of them there are approximately 10 bases that are present. Okay, so there are 10 base pairs in each of the turn. Okay, in each turn you see that there are 10 base pairs. Okay, and the distance between each one of them, one of the bases, is 0.34. Okay, so basically you see that this is how the DNA structure is. Okay, and you see that the plane of one base pair stacks over the other on the double helix, and yes, this is what confers stability to our DNA. Okay, looks like very simple, but yes, to get this structure there was lots and lots of work that had to be done in the field of molecular biology, and all this was done quite a lot in our DNA molecule. And yes, how this DNA gives rise to a protein would be our topic for our next class. So I hope you have got a thorough idea of what DNA is and how this DNA works, um, what is its structure and all of them. Very important with today's class is the components of the nucleotides, then how they are linked and all that. So more than trying to remember it through structurally, I would say imagine it visually, so that it's something like easier for you to remember all these structures, right? So yes, so let us just wind up with the class. In our next class we will talk more about how this DNA becomes a chromosome, right? And we will also talk about one very important part of molecular biology, which is called as central dogma of life. Fine. So see you again in the next class.